a TB vaccine because TB is a global disease. It causes a lot of health challenges. It burdens the healthcare system. And as many people say, prevention is better than cure. So a TB vaccine would prevent people from actually getting sick, and that is actually the aim of what we're trying to do. It is essential to actually know which part of TB you want to incorporate in a TB vaccine, because the bacteria that causes TB has many different proteins, and we want to actually know which parts are the Achilles heel for TB. So you really want to get an understanding of which of these proteins that TB makes do you want to target with the vaccine. Now this has been very challenging because there are many potential targets, and the project that I'm going to talk about really tried to get an idea of which of these targets should we put into a new TB vaccine. The ACS study really collected samples that are essentially a gold mine, and it's actually very interesting that this particular study actually started before I joined SATV, and I think it's a testament to the efforts of other people who came before me to build these interesting and unique sample sets. Now, the ACS cohort was a cohort where we followed up adolescents for 2 years, collecting samples about every 6 months or so. And because we're able to do this, we could then have a very nice data set that could allow us to ask very interesting questions about TB progression. And in this case, in the study that we did, asking, are there certain proteins that people who do not progress to TB disease recognize that maybe other people who do progress do not recognize? Now, specifically for this study that we worked on, we collaborated with individuals at Stanford University, as well as individuals locally at Wits University. And I think these collaborations allowed us to leverage the expertise of researchers both at Stanford as well as Wits to be able to come up with a candidate vaccine for TB. So the Titan project really comes out of this data from the adolescent cohort. And again, this is the cohort where we had samples from individuals who controlled disease and individuals who unfortunately progressed to TB disease. And we're able to identify, using these samples, proteins that we've incorporated into essentially a candidate or a novel TB vaccine. And this vaccine contains 4 proteins that TB makes. And we've been able to put these 4 proteins in the form of an mRNA. And using this mRNA, we're now in the beginnings of a product development project. Now, this entails us making sure that the vaccines that we're making induce immunogenic responses, or induce responses that we think are protective. We're also testing it to see, if mice are vaccinated, do they become better protected than unvaccinated mice? And then we're also working with Afrigen, which is a local partner, to look at can they actually manufacture this, and are there any issues of toxicity? So it's essentially the beginning of a product development project. So the Titan project is very exciting on many fronts. I think from a research point of view, I think it's very exciting that we're able to follow up some of this work that we've generated and really push the boundaries. But we're also able to generate a lot of capacity in terms of vaccine development. And what the Titan project allows us to do is essentially demonstrate that we can discover interesting antigens in South Africa with South African collaborators, develop and design a novel TB vaccine. And the end goal is essentially to then see if we can actually manufacture it in South Africa, and therefore have the entire pipeline of discovery to development and eventually manufacturing. Coming up with the list of antigens that we found was actually a very interesting and exciting process. So, as I've mentioned, the ACS study was this large study where we had samples collected for many, many years. What we then did was we're able to then go back to these samples and retrieve them. And we're able to stimulate the cells in such a way that we're able to identify cells that would respond to the bacteria that causes TB. And once we did that, we're then able to sort each of those cells individually and actually ask the question, which part of the TB bacterium are these cells recognizing? And we were specifically looking for differences between cells that came from individuals who controlled infection versus cells that came from individuals who progressed to TB disease. Now, once we had identified those cells that were enriched or more abundant in people who controlled infection, we then asked, so which part of TB do they recognize? And I think it was quite exciting when we actually were then able to identify the parts of TB that these cells that were enriched in people who controlled infection recognized. And when we had those, we're then able to then put them essentially into an mRNA vaccine, so encode the sequence that allows the body to make these proteins, and therefore the body is actually able to make the vaccine in real time, which was pretty exciting.